Hi, I'm Conor Rotten. This is the second half of the computational section of PHPH20007. So this will just be one lecture, whereas the first half had two lectures in it. And this lecture will be about a different approach or a different attitude to modeling in the brain. If you remember, in the first part, in the first two lectures, we discussed detailed models, the integrated and fire model, and leading on to things like the Hodgkin-Huxley model, which are actual simulations of the dynamics of the neurons. And the belief there is that we can make a detailed simulation of what's happening in neurons or in a neuronal network, and that can be compared to experiment, and that can allow us a more exquisite probing of those experimental results, because they can relate what we can measure, spikes or whatever, to what we'd like to measure, synaptic dynamics, for example. Here, we're going to take a much more top-down approach. We're going to try and understand in a model how some aspect of computation in the brain might occur. And there, our goal might not be the detailed comparison of spike times or voltages that occurs in the more low-level approach, but rather a qualitative agreement. We can see that the sort of thing that is happening in the model compares well with the architecture, the cellular arrangement, or some other properties of the network that we are examining. And so the idea here is more to answer why questions. Why would the brain be doing what it's doing? Or how questions. How is the brain doing calculations? How is the brain doing the calculations it needs to do to support cognition? And of course, the idea eventually would be that these things all kind of join up in one big picture, and you can have an understanding of everything that's happening from the cells up to the whole brain. And of course, this in the small case, in the detailed case, in the low-level modeling we discussed in the first two lectures, it is still kind of about understanding. When you simulate something, when you model something, you kind of understand it because you're able to compare it to something that you have complete control over. And of course, the experimental, the comparisons you do there will never be precise, and in this case, they're just not quite as precise. But it does, the two examples that we're doing, the integrated fire model we did the last time, and this more high-level model we're doing now, do kind of represent different attitudes or different scales of modeling. So in this case, we're introducing a more top-down style of modeling, and we're going to do it by looking at the hippocampus. This, of course, is a picture of the hippocampus. It's that funny seahorse-shaped part that we find at the edge of the cortex, near the middle of the brain, where the cortex comes back under itself. What's it for? Let's talk about that first. What's the role of the hippocampus? Well, the hippocampus is extremely well studied. It's one of the most studied parts of the brain over the last 30, 40 years. There's a couple of reasons for that. It is quite amenable to in vitro recordings. You can slice it in a way that preserves a lot of the connections. It, of course, was the brain area that was most important during the birth of in vivo recordings. It's a part of the brain that's implicated in significant diseases with a significant burden, such as Alzheimer's. And I think most of all, it's a part of the brain where we feel that we can map all the way from looking at what the individual neurons are doing to understanding the behavior of animals. And of course, that's because the hippocampus is known to be important for memory. It's not just important for memory, it might play a role, for example, in smell, but it mostly seems to be about memory. And there are two stories told about the hippocampus and memory. These aren't rival stories. They're presumably part of a single larger story. But they are still, as far as we're concerned today, slightly different stories. There's one about place fields and place memory, and that's, of course, the thing that's best studied about the hippocampus. And then there's a second story, which is about auto-associative memory, and that's the story that I will tell today. And so this story is about a type of memory that's different from the working memory. That's the memory where somebody says to remember 7 numbers, and they go round in your head. And so there's ongoing electrical activity rather than synaptic changes that allow you to continue to represent that information. Then there's the very long-term memory that we use when we're cogitating, the memory that fabulously allows us to go from one thing to another, that associates something we see with something we remember, something we read in a book, something somebody told us, some other fact we have. The memory that we use as the basis for our imagination and our scientific ability, our ability to write novels, etc. Then there's another sort of memory, which is, again, another type of declarative memory, another sort of memory that you can put into words, which is how we just remember something for a few hours or a few days. And we don't put it into memory by thinking about it an awful lot or having lots of experience with it. It goes into memory after only 1 or 2 exposures. It's the memory where somebody tells you their name at the start of a meeting, you remember their name. I mean, not me, but other people might remember that person's name when they meet them later at the coffee table. It's the memory where you go out in the evening, you cycle to when you're going out. Later on, you don't feel like, or you're not in a position to cycle home. So then the next morning you have to go and get your bike. So you have to remember where you locked it. It's that kind of memory. It's a sort of medium-term memory. It's not a memory that lends itself to this kind of thinking, where you go from thing to thing. That's is thought to take place. That sort of memory is thought to be stored in the cortex. This hippocampal memory is where memories are supposed to go first, and it's you remember things quickly and you recall them quickly. So how does the hippocampus do that? Well, let's just look at the hippocampus for a second. The hippocampus has, I'm going to ignore the subiculum, so let's say it has 2 parts. It's the cornu ammonis, which is the horn of Ammon. It's supposed to be shaped like the horn of Ammon, which is an object in Egyptian mythology. The horn of Ammon is divided into 4 parts, although we don't really talk much about CA4, which is actually also called the hilus. Sometimes it's considered part of the dentate gyrus, and the CA2 you do hear it mentioned, but it's a little part in between CA1 and CA3. So mostly the CA is divided into 2 parts. CA1, which is implicated in place memory in memory fields. We won't be talking about in CA3, which is really important in the story that we're discussing now for associative memory. The dentate gyrus is also part of that story. It's supposed to do pattern separation. We won't talk about it much today, but we will return to it at the end. But there's a fuller version of the story puts a lot of emphasis on the dentate gyrus. Here is the dentate gyrus. You can see the cells, and we'll return to this, are different. CA3 has these large pyramidal cells. Dentate gyrus is psychologically distinguished from CA in that it has a different type of cell, the granular cells. This is the entorhinal cortex. It's not formally part of the hippocampus, it's part of the perihippocampal region, but it is important in the hippocampus in that the information flows to the hippocampus from the entorhinal cortex, and information comes from the hippocampus again to the entorhinal cortex, albeit at different layers. And so when we discuss the hippocampus, it's typical to include EC as well. This is the connectivity. As you can see, stuff goes either directly to CA3 or to CA3 roundabout via dentate gyrus. We will mention this, although we won't talk about it much. And then via CA1, it comes back to the entorhinal cortex. And this little loop here is to emphasize the fact that CA3 has a lot of recurrent connectivity. So these are the cells, the dentate gyrus. This layer here is for the adult animals. P is the number of days since birth. You can see that it has these granular cells. They have a reasonably simple connectivity. We talk about the dentate gyrus as being a feed-forward network. So that's a network layer where the nodes in the network, in this case neurons, aren't connected to each other. So stuff comes in and comes out, but there's no connection within the layer. It's not literally true of the dentate gyrus. It does have interneurons that connect from granular cells and to granular cells, but the principal cells, the granular cells, don't have connections among themselves. There's also a feedback loop with the hilar. There's these large cells, the mossy cells, that cause feedback. So again, it's almost always a simplification when you say a network in the brain is a feed-forward network, and it's certainly a simplification here, but it is a common simplification. CA3, in contrast, is highly interconnected. So it has these large pyramidal cells. You can see the complexity of their dendritic arbor. They have basal dendrites, apical dendrites, lots of them, and the principal cells are connected to each other. You get connections directly from one CA3 pyramidal cell to another, and that will be important in the story we're telling. So, auto-associative memory, that's the computer science, as it were, model that we're going to try and map to the CA3. So in auto-associative memory, and indeed in any picture of memory, the memory is made up of a pattern of neuron activities. Here in this picture, and we're going to return to this idea again, the neurons, well, they're just on-off neurons. So a neuron is either firing at its low rate, some basal rate, some background rate, represented by these empty circles, or it's firing at its high rate, represented by these red circles. And so this is the low rate neurons are off, the high rate neurons are off, so on. And so each neuron is described just by a single number, 1 or 0, corresponding to whether it's on or off. And so that's a lot simpler than the spikes and voltages, etc., that we use normally in describing neurons. So, well, how is a pattern a memory? Well, that's just these individual neurons would represent part of the memory, and they'd come together to code for the whole memory. And then subsequent neurons in CA1 and then in entorhinal cortex will be able to read off these memories. If particular neurons are all active together, then a neuron that these all neurons connect to will become active as well. And that would constitute the recognition of the presence of that pattern. So the memories are patterns of neurons. In a sort of simplistic thing, you could imagine, you've been told about a penguin for the very first time. You set up a pattern of neurons corresponding to penguin. This first neuron that's red would be maybe a black and white bird. This next neuron that's red would be a swimming bird, flightless swimming bird, and this last neuron that's red corresponds to the word penguin or whatever. Now, obviously the little bits that the neurons code for much smaller pieces of the information, and the story is much more complicated, but that's a cartoon that you can keep in mind. And then by an auto-associative memory, we mean a memory that allows patterns to be completed. So the CA3 has lots of interconnections between the neurons. So these nodes here will have connections between them. And so when some of the neurons are active, if these two neurons, for example, have strong connections to this neuron here, well then when these two neurons are active, they cause this neuron to become active as well. And maybe there would be negative connections with the other neurons. So when these two neurons are active, they'd stop the other neurons becoming active. And so what would happen then is if the input, which is, as we've looked at, coming from EC, from entorhinal cortex, lights up these two neurons, then the internal dynamics of the layer will light up this neuron, completing the pattern. So somebody mentions penguins as a black and white bird, and that allows you to remember that penguins are flightless. And that dynamics is the intrinsic dynamics of the network itself. So the question is, can we design the network so that it can store memories in that form? And as I said, it's made of off and on neurons. These were first discussed by McCulloch and Pitts, working kind of at the dawn of the computer age, just after the Second World War. They thought that based on what they knew about biology, this would be a good element in designing computers. And it's part of the long story of interaction between computer science, artificial intelligence, machine learning, and neuroscience, where ideas about neuroscience are used in thinking about how machines might learn, and then looking at how that machines might learn gives you ideas about how computers might work. So this is the McCulloch-Pitts neuron. It's off and on, and then, as I said, these neurons are all connected. So here it's drawn as an all-to-all network. Each neuron is connected to every other neuron in the layer. That wouldn't be true. I mean, the CA3 is a highly connected network, but it's not connected with so that every neuron is connected to every neuron. But this story carries through for more sparsely connected networks quite easily. We just won't go into those details. But we will give a name to the different connections. So the idea is that the connection from neuron 0 to neuron 7 is called W07. The connection from 2 to 8 is called W28. They're just two that it's easy to fit in the name because of the lines everywhere, but it doesn't matter. Any connection is labeled by the starting and end neuron. And the important thing, and really a gross simplification relative to what happens biologically, is that these connections are symmetric. So the connection from 0 to 7 is the same as the connection from 7 to 0. So W07 is actually equal to W70. And we'll see that the learning rule that we have for changing or adjusting these W's is symmetric in the two numbers. So again, that's not at all like real neurons, but it's useful for this approximation. The other thing is that the W's aren't constrained to be positive or negative. They can be either, and that is completely unlike real neurons, where a given neuron either is excitatory and has positive connection strengths with other neurons, or is inhibitory and has negative connection strengths. You can see how you could get around that. You might have interneurons, so to make negative connection strengths, the connection goes via an interneuron, and that kind of might explain why interneurons have local connectivity, whereas in many parts of the brain, whereas excitatory neurons have more distal connectivity. However, that's not we're not staking ourselves to any particular description about how this approximation might be fixed here. We're just saying in this simplified picture, these W's can be positive or negative. And so here's the learning rule. This is a Hebbian learning rule, so it's based on Hebb's idea that neurons that coincident activity between neurons strengthens the connection between them. But what Hebb said was actually a little bit different. He said if A causes B to fire, then the connection from A to B is strengthened. But here we don't really have any causality because we don't have spikes, we just have neurons being on and off. And we have symmetric connections, so we can't say the strength of the synapse from A to B. So this is a sort of cartoon version of Hebbian. It's of Hebb's law. It's the neurons that fire together, wire together version of Hebb's law, which we know is not an accurate description of how neurons work, but it is enough of a close enough for auto-associative networks to work. So the idea, again, is that we have some connection strength. Delta just means change. The change that will be made to that delta, to that W, ij, the connection strength from I to j, will be some small number eta. You want to learn fast, but not too fast, or else the connection strengths will be too vulnerable to noise, or be too easy washed out by subsequent activity. And then we have xi is the on or offness of the ith neuron. So if the ith neuron is on, it's a 1. If the ith neuron is off, it's a 0. The same for the jth neuron. And then the A, well, A is just some number that is supposed to tell us the average connectivity of the it tells us the average activity of the network. So as you can see, I just corrected a small typo in that space. So basically, A is the fraction of neurons that are on in a typical pattern, in an average pattern. So in the examples that we've been looking at, we only kind of draw 8 neurons, so to make something that looks like a pattern, we have to use up quite a fraction of them. But you imagine the hippocampus has an awful lot of neurons. In our brains, the hippocampus isn't such a significant part of the brain, but in the rat's brain, it's a big part of the rat's brain is the hippocampus. It does have lots of neurons, and so you wouldn't expect too large a fraction of the neurons that could be on to be on in a given memory to represent a given pattern. And so this A is some smallish number, and as I said, it represents, you can see we're not going to prove this, but in the dynamics that we're talking about here, if you make these changes to the neurons, the number of neurons that on average are on for the patterns will eventually end up near A. So this is this so-called sparseness parameter. Either way, let's have a look at what happens if we've got 2 neurons connected. So here we have 2 neurons connected, and both neurons are off here. And so the xi and xj, in this case x0 and x1, are both 0. Minus a by minus a is a squared. And so a is a small number, eta is a small number. And so this change in the synaptic strength, connection between these 2 neurons, is a small positive change. So off and off causes a small increase. Off and on, in contrast, causes a decrease. So we have, in this case, the 0 neuron is off, so that's a 0, so that's a minus a, you can see there. And then this neuron is on, so that's a 1 minus a. A is a small number, so 1 minus a is positive. And so this number is bigger than the number we had before, eta a squared, because 1 minus a is bigger than a, because a is very small. But it's a decrease. We have a negative sign there. And so if one neuron is on while the other is off, the connection strength between those 2 neurons gets smaller. And then in this case here, where both neurons are on, now this isn't a big increase because the idea is that eta is a smallish number, but it's bigger than the 2 previous changes. And so if both neurons are on together, well, we get 1 minus a squared by eta. There's a positive, it's a plus there, and so the connection strength between the 2 neurons increases. So that's a so-called correlative law. It's Hebbian learning for one of these on-off neuron networks. And so the idea now is that when you're learning the pattern, your entorhinal cortex switches on some number of the neurons corresponding to the pattern. Black and white, swims, doesn't fly, cold penguin. And as a result, the connection between those neurons gets stronger, and the connection between these neurons and the other neurons, well, it gets weaker or becomes negative depending where it's starting off, but it's changed in a negative way. And so that's what happens during the presentation of this pattern. And so the Hebbian learning builds into the network these particular connection strengths that are connecting together positively or in a stronger way, the neurons that are active together during the pattern, and those neurons are connected in a weakly or even negative way to other neurons. And now, what about the activation? What is it that causes the neurons to come off and on? Well, I don't want to go into too much details here, but the idea is that the input of each neuron, so each neuron has an input, which we'll call HI here, which corresponds to the weighted sum of all the other neurons, and they're weighted by the connection strengths. So this symbol here, the sigma, is a sum, and so HI is equal to WI0, so this is the ith neuron, there's a contribution from the 0 neuron, WI0 x0, so that gives you WI0 if x0 is 1, and 0 if x0 is 0, plus WI1 times x1 plus WI2 times x2, and so on. So the input to a given neuron is the sum of the connection strengths to that neuron from all the other on neurons. So all the other neurons that are on contribute to the HI for the neuron that you're considering, but they contribute weighted by the synaptic strength. And then if that HI is bigger than some theta, so theta is a parameter, it's some number, changing theta changes the activation of the activity of the network. So we're not going to discuss that here. But if the HI is bigger than theta, then that neuron comes on. Xi is set to 1. Otherwise, it's set to 0. So you can see now how this works. So in this case here, where now the entorhinal cortex presents, there's input to these neurons as well from the entorhinal cortex. So the entorhinal cortex input switches these two neurons on. There's not enough input from the entorhinal cortex to switch any of these other neurons on, but the internal dynamics, the auto-associative dynamics now mean that that this neuron is receiving W, whatever this is, 225 times this one, and W02 times this one here. And during the learning phase, back here, we saw that these connections had become strong, and so that gives this neuron plenty of input, and that switches it on. So the associative learning now trains the W's, strengthens some of the W's, weakens the other W's to allow for this auto-associative dynamics to happen. In other words, during the training, you're switching on some of the neurons, switching off some of the others. That changes the synaptic strengths, and then later on during recall, if you present a part of the pattern, the dynamics will fill in the rest of that pattern. Now, obviously, this isn't going to, allow you to remember everything. And you can see here that there's some sort of, if you're trying to remember two memories, there's going to be some sort of competition. So if you're remembering the top memory there, you can see the connection from 1 to 5 is going to be weakened in order to remove any connection between 1 and 5 to stop 1 lighting up when 5 does, basically. But in this pattern, another memory, this other memory also includes 5. So 5 is the word penguin. So in this memory, you're trying to remember the chocolate bar penguin. So 6 corresponds to chocolate and 1 to biscuit. Well, then there's a confusion, because here now you're increasing the connection strength between 1 and 5. And so there is a limit to the capacity of this network. At a point at which it's going to start getting confused. It will complement flightless bird, penguin, with biscuit, and that would be confusing. However, this is something that people can study. There's lots of neurons. There can be some overlap because, as I said, the patterns are made up by more than 3 neurons. These attributes would be split into smaller attributes, and so some overlaps are allowed. In fact, there's a kind of a hand-waving argument that says that you have n-squared connections. So you have n neurons. So in fact, you have n multiplied by n minus 1 over 2 links between these neurons. So that's a bit like n squared. There's every neuron, every pattern, you have to decide for each neuron which, whether it's a 0 or a 1, whether it's on or off. So there's n neuron values that have to be given for each pattern. And so that sort of says that n-squared numbers can be, you need to fix n of the n-squared numbers to get a pattern, and n squared divided by n means that you can store n patterns. Now, that's just a rough order of how much you can store. In fact, it would be some fraction of that n. But the more neurons that you have, the more patterns you can store, and it kind of goes linearly. So that's the hand-waving calculation of the capacity of a network. There is an additional problem with this, which is that, in fact, often networks are, are sort of overlapping. It's our memories are overlapping. It's not simply that you have random memories and you're storing them, and sometimes they overlap too much, and if you try to put too many in, you start getting confused. It's also the fact that many of the things you might remember or be trying to remember will have things in common. So, maybe you're remembering stuff about birds. Well, lots of these birds will have this attribute here of having wings. And so that's the attribute of having wings. This is the attribute of being able to fly. And this is, the first line corresponds to an eagle, 5 corresponds to an eagle, 6 corresponds to a robin, 7 corresponds to a thrush. And then 8 corresponds to a penguin, and of course it doesn't have being able to fly, but it does have being able to swim. And so this is very confusing for the network, because there's lots of instances where it's learning a strong connection between 0 and 1. And so you might think that when it's if it's presented with some attributes of the penguin and the first thing, it might fill in not just being able to swim, but being able to fly, and then you'd mistake the penguin for an auk. Or even just might fill in being able to fly, because it's had so many occasions to learn a strong connection between 0 and 1, and much fewer occasions to learn a strong connection between 0 and 2. And, well, there is a thinking about this, and I'm not going to talk about it in detail, but the idea is that the dentate gyres fixes the problem. The idea is that the memory isn't stored directly in terms of attributes, but rather there's some randomization that occurs, and that the dentate gyres, that the CA isn't storing memories so that things that are similar to each other are similar to each other. Putting these memories in the same sort of place would mean putting all the birds together. If you were using the same patterns, same sub-patterns for different types of birds, it would make it easy to go from one bird to another. And you could think of this pattern completion as kind of a thought process. Penguins, wings, they can swim, but can they fly? Well, what birds can fly? By putting the memories together, you would be allowing yourself to drift from one idea to the other. But that's not what CA is for. That's not what hippocampal memories do. They try not to confuse things, but try to separate them out. And so the belief is that the dentate gyres provide some sort of random recoding of the memories, so that even similar memories will actually end up being corresponding to very different patterns. And so you as you are trying to remember the memory, you recode it randomly, and then the CA2 or 1 or something sorts that out and does the decodes the memory. So in this picture, the dentate gyrus is there just to sort of randomly scramble up your memories. It's still important, as long as you have a translation there, it's still useful to be able to complete the patterns. But the tokens in the pattern don't directly correspond to the attributes. They've been translated from the attributes into some sort of random set of tokens, and then those random set of tokens are translated back into the attributes. And so there is an idea that pattern separation is important. Patterns separation, as I've actually written there. Pattern separation is important for preventing the collision that occur when you're trying to remember things that have overlapping or similar attributes. So, in summary, CA3, many recurrent connections. The excitatory neurons are connected to each other. CA3 is thought to be an auto-associative memory store. Patterns are completed by its dynamics. The capacity is proportional to n. Dentate gyrus is feed-forward. There's no lateral connections between the excitatory neurons, and its role is to separate patterns ready from the EC, ready for storing in CA. So, as I said, this is quite a vague model. We can fill out more details if you read more about it, but it does give you a picture of what might be happening, and really the sort of provocative thing is that it requires CA3 to be highly recurrent, which CA3 is. You can either try and match up other aspects of CA3, like the sparseness of the firing, etc., or in the case of dentate gyrus, which performs its randomization function, the fact that the dentate gyrus has a neurogenesis adult neurogenesis is one of the few parts of the adult brain where new cells are born, might be something to do with it as well. Excellent. Thank you very much.